Hello students, welcome on board. This is part 2 of the chapter Sexual Reproduction in Flowering Plants. Here we will be covering the female reproductive unit, the process of Megasporogenesis, and the embryo sac. Without further ado, let's begin with the first topic. Now first we learn about the female unit of a flower. The female unit of a flower is called pistil, or it is also called as carpel. Now, a wall of pistil is known as gynoecium. It is known as gynoecium. So, a female unit of a flower is called pistil or carpel, and a wall of pistil is called gynoecium. Okay. Now, if there is only one pistil, the condition is called monocarpellary. It is called monocarpellary. And if there are multiple pistils or more than one pistil, then it is called multicarpellary. It is called multicarpellary. Now, if the pistils are fused, then it is called syncarpous. And when the pistils are free or separate from each other, then it is called apocarpous. Alright? So if there is only one pistil, then it is called monocarpellary. If there are multiple pistils, then it is called multicarpellary. If the pistil is fused, then it is said to be syncarpous. And if the pistils are free, then it is called apocarpous. Alright. Now let us see the parts of the pistil. The pistil has mainly three parts, and they are stigma, style, and ovary. Alright? The stigma is the region that receives the pollen grains. It is the region that receives the pollen grains. And style is the slender part that is found below the stigma, and ovary is the bulging part at the base. Now we are going to learn the structure of an ovary with the help of a diagram. Here you can see the structure of an ovary. Within the ovary, there are cavities which are called locules or ovarian cavities. Now inside the cavity, there is a region that is called placenta. The circular region that you see here is called the placenta. And then from the placenta arises ovules or megasporangia. The white grain-like structure you see around the placenta, these are called ovules or megasporangia. Alright. So within the ovary, there are ovarian cavities or locules. Inside the ovarian cavity, you will find placenta, and from the placenta arises ovules or megasporangia. Now, these ovules are attached to the placenta with the help of some stalks which are called funicle. So, ovules are attached to the placenta with the help of a stalk that is called funicle. Now, the junction between the funicle and the ovule is called hilum. Hilum is the junction between the ovule and funicle. Alright. Now, when you look at the second figure, you will see that the ovule is covered with the help of two protective layers, and these are called outer integument and inner integument. Now, the region where the integuments are absent, this is called micropyle, and opposite to the micropyle, you will find the chelacal pole or chelaza. Now, within the integuments, there is a mass of cells which is called nucellus. And the nucellus contain food reserves that nourishes the embryo sac or the female gametophyte. So we've learned that the ovule is protected by two integuments, the outer integument and the inner integument. Right. At the micropylar end or at the micropyle, the integuments are absent. Integuments are absent. And opposite to the micropyle, there is a chalasil pole or chalasa. We've also learned that within the integuments, there is a mass of cells that is called nucellus, which contains food reserves that nourishes the embryo sac or the female gametophyte that is present within the nucellus. Alright, so we've learned that within the ovary there are ovarian cavities or locules within which there is a region called placenta that give rise to ovules or megasporangia. Now these ovules are attached to the placenta with the help of a stalk that is called funicle. And the junction between the ovule and funicle is called hilum. Around the ovule, you will find two protective layers, an outer integument and an inner integument. And the region where the integuments are absent is called micropyle, and opposite to the micropyle, you will find the chalasal pole or chalasa. Now within the integuments, there is a region that is called nucellus that contains food reserves which provides nourishment to the developing embryo sac or female gametophyte. Now we will learn the process of megasporogenesis. Megasporogenesis. Megasporogenesis is the process of formation of megaspores from megaspore mother cell. So, the megaspore mother cell give rise to megaspores. And this process is called megasporogenesis. Megasporogenesis is the process of formation of megaspores from the megaspore mother cell. Alright. Now let's just see what happens in the process of megasporogenesis. Now we know that there are ovules within the ovary. These ovules differentiate. They differentiate to form the megaspore mother cell. Alright. Now the megaspore mother cell then undergoes meiosis to form 4 megaspores. It forms 4 megaspores. Now one megaspore remains functional and the remaining 3 degenerates. One megaspore remains functional and the remaining 3 megaspores degenerate. Alright, now this functional megaspore then finally develop into the female gametophyte or embryo sac. So the development of a female gametophyte or embryo sac from a single functional megaspore is called the monosporic development. it is called the monosporic development. All right. So we've learned that the ovules in the ovary differentiate to form the megaspore mother cell, which undergoes meiosis to form 4 megaspores, out of which 3 degenerate and 1 remains functional. And this functional megaspore then finally develop into female gametophyte or embryo sac, and this development is called monosporic development. All right. Now let us see in detail how a functional megaspore develops into a female gametophyte or an embryo sac. First, the megaspore, that is the functional megaspore, undergoes mitosis to form a 2-nucleate embryo sac. It forms a 2-nucleate embryo sac which further undergo 2 more mitotic divisions to form a 4-nucleate embryo sac, and then finally an 8-nucleate embryo sac. 8-nucleate embryo sac. So the functional megaspore undergoes mitosis to form a 2-nucleate embryo sac, which further undergo 2 more mitotic divisions to form a 4-nucleate embryo sac, and then an 8-nucleate embryo sac. So the embryo sac has 8 nuclei, which is why it is called 8-nucleate embryo sac. All right. Now we know that an embryo sac is 8-nucleated, right? Now within the embryo sac, 6 nuclei get enclosed by cell walls and they are organized into cells. All right? They get organized into cells. And the remaining 2 nuclei are called polar nuclei. Now the cells at the chelastral end is called antipodals, and at the micropylar end there are 2 synergids and an egg cell. And together it is called the egg apparatus. It is called the egg apparatus. All right. Now if you see the polar nuclei, it is enclosed within the central cell, right, which makes it 7-celled. Therefore, we can say that the embryo sac is 8-nucleated and 7-celled. So a typical angiosperm embryo sac is 8-nucleated and 7-celled. So that's it for the session. This was part 2, where we've learned about the female reproductive unit of a flower, the process of megasporogenesis, and formation of embryo sac. In part 3, we'll be covering pollination and different types of pollination. If you found this helpful, please do like the video, and if you have any doubts, comment down below. Subscribe to watch the upcoming videos. Thank you for watching.